animation, you can see that if G is linked to A or T instead of C, it cannot maintain the 3D structure of the DNA molecule, and it can cause to mutation, DNA mutation. And DNA mutation can be benign or sometimes can be harmful, depend on the position of the mutation. And we will study more detail on this on chapter 4, DNA genetic variation and genetic mutation. Okay. Normally, most of the DNA mutation are benign because as we explore the human genome, 98.5% of the human genome is not coding for protein. It's sometimes part of it is reputation. Part of it is control genome region, which is control the expression of the other genes. Only 1.5% is coding for protein. And if it happens, the mutation in this region, it could be critical for the carrier. But otherwise, it would be fine. We will discuss more on this on chapter 4, DNA mutation. So we come to another aspect is the differences in the structure of DNA and RNA. And as you can remember on the survival kit, yeah. So in survival kit, so this is a important part you want to remember if you want to pass this course with minimum results. So in chapter 1, you have to focus on the structure of a nucleotide. So we have discussed already on this. So structure of nucleotide is composed of 3 main components: a 5-carbon sugar. In case DNA is the deoxyribose. In case of RNA is ribose. Base nitric, there are 4 types: A, C, G, and T, and DNA and A, C, G, and U in case of RNA, and a phosphate group. Okay. The second one is structure of DNA and RNA and the differences between the structure of DNA and RNA. So this is the part that you should focus on. So in a nutshell, there's several main differences, fundamental differences between the in the structure of the DNA and RNA. First is 5-carbon sugar. In DNA, we have deoxyribose, that means lack of one oxygen atom in the carbon number of the hydrogen group in carbon number 2. Remember, in RNA is ribose. In the basin, in the nucleotide, we have DNA, we have A, C, G, and T, and in RNA we have A, C, G, and U. Uracil in RNA instead of T in DNA. In the structure of the DNA, in DNA we have most of the molecule of DNA is double strands, and in RNA, even some RNA molecule in double strand, especially in the genome of some viruses. Most of the RNA strand is single strand. DNA was used as a genetic material for most of the living organism, eukaryotic cell, prokaryotic cell, and virus. Meanwhile, in RNA, it used as a genetic material for only some virus. Because normally the RNA was less stable, so to store and pass into information, DNA is much more convenient and stable to protect the code of life. For the function, DNA, of course, is to replicate and store genetic information in the nucleus, and in RNA is the mRNA is transfer the genetic information from DNA to protein. Meanwhile, we have also other type RNA like ribosome RNA in the structure of the ribosome, tRNA to transfer the amino acid from to synthesize the polypeptide, We have also other RNA like long non-coding RNA, or sometimes we call linked RNA, and short non-coding RNA, which is play role in the epigenetic. We will discuss on that on chapter 7. In terms of structure, DNA is much more much more stable than RNA because it was composed by 2 double strands. So because of the complementary link, so it's be more stable than RNA. Meanwhile, RNA, because most of RNA is string strand, so it's less stable. And also of the hydrogen group in the carbon number 2, which is easy to cleavage by the hydrolysis reaction. And in terms of length, normally DNA is much longer than RNA. Okay. So we come to the next part is the genome of a virus. So the virus is composed, the structure of the virus is quite simple. So simple that some scientists are not consider a virus as a living organism. Because a living organism must be characteristic by ability to metabolic mechanism. Meanwhile, the virus cannot produce energy by themselves. They must based on the system and the material of the host. But anyway, the composed of a virus is composed by a nucleus. The nucleus can be DNA or RNA, can be single strand or double strand, and the envelope of protein. So because of the simple structure, so the size of the genome virus is quite relatively small. So the size range, it comes from 2,000 base pair to 2 million base pair. So just to compare numbers, in human, we have the human genome the size is about 3 billion base pair, 3 billion base pair compared to. So this is the structure of 2 very common virus, RNA virus, which is the virus have a nucleus is composed by RNA. So we have the HIV virus and we have coronavirus. HIV is causing the AIDS disease, AIDS, and the coronavirus causing the COVID-19 pandemic that we still have to suffer nowadays. So let's come to the infectious pathway of one virus is HIV. So in the HIV virus, in the protein envelope, it have a receptor called GP120. And the HIV is in fact only in one cell type, which is the T4 leukocyte, T4 white blood cell. So in the membrane of the T4 leukocyte, it has a receptor called the CD4 receptor. And the HIV recognize the T4 leukocyte based on this receptor CD4. When the receptor of the virus GP120 recognize the CD4, it will activate its invaded form and start to inject its nucleus material, which is on a molecule here, inside of the host cell. And this RNA using the material and enzyme, using the material of the host cell through enzyme called RNA reverse transcriptase or rtRNA amylase. This enzyme will synthesize the DNA based on the sequence of the RNA. So in the consequences, we will have a DNA molecule which have the sequence complementary match with the RNA molecule. So this DNA will match with the DNA nucleus of the host and start to synthesize new RNA. And from this new RNA, the ribosome will start to synthesize the protein envelope in the beginning. So as a result, from one cell, from one virus cell, the T4 can replicate thousands of HIV virus from here, and it starts to release out of the T4 leukocyte and invade it into the other cell. So next, we will come to the prokaryotic genomes. A prokaryotic genomes, firstly, is a DNA molecule, not like in some virus is RNA. First, from prokaryotic to eukaryotic, we have the nucleus was DNA, okay, the DNA molecule. And in prokaryotic cell, the DNA is circular. That means it's not linear DNA, it have no ends. Is composed like a circular. In some prokaryotic cell or some bacteria, we have a small addition circular DNA. For example, in here we have the nucleus DNA was marked in red here, and we have the extra DNA called plasmid. This plasmid play a critical role to help the bacteria having ability to again some kind of several types of antibacterial medicines. We'll talk about it in chapter 3. And the DNA molecule in the prokaryotic cell was attacked with several nucleus protein to protect any damage into the DNA molecule. And it has two shapes. The shape of quite relaxed here is relaxed DNA is quite it not twist at all. And in the other shape is supercoiled DNA that you can see that the DNA fragment is it was twist maximally for easier to divide in cell cycle and easier to protect. But in the supercoiled state, there's no transcription factor or also RNA DNA polymerase complex can come and binding into the DNA to start to replicate the DNA or transcribe the DNA, because those enzyme complex need space to binding into the DNA. So it can only bind when the DNA molecule was on the least relaxing state. And here is electrophoresis of the DNA plasmid. For example, we put a negative charge and positive charge in here. So because you remember that the DNA was negative charge in the neutral pH medium, so it will moving from the negative charge to the positive charge, and based on the distance of the moving of the DNA fragment, we can identify the size of the DNA molecule. So in here, even the DNA have a different size, but based on different state. One is supercoiled, that means it's twist maximally, and the liner, that means it's in the liner state, and the nickel circle. The supercoil is moved the fastest, and meanwhile, the liner and nickel circle was moved slowly during the electrophoresis. So let's watch an animation on how a genome in bacteria was structured.
The circular chromosomal DNA of bacteria must be compacted about 1,000-fold to fit within a bacterial cell. Part of this compaction comes through the formation of loop domains, where segments of DNA are folded into loop-like structures that are held in place by DNA-binding proteins. The formation of loop domains compacts the DNA about 10-fold. Further compaction is accomplished through a process known as supercoiling. In this process, which is under the control of the enzymes DNA gyrase and topoisomerase 1, twists are introduced in the DNA molecule that cause it to compact by coiling onto itself. This is much like the process of coiling that you can observe by twisting a rubber band. DNA looping and supercoiling make the bacterial chromosome more compact so that it can fit within the nucleoid of the bacterial cell.
So come to the eukaryotic genomes. So in eukaryotic genomes, we have the DNA nucleus was located inside the region called nucleus, with the nucleus membrane separate the nucleus with the other part of the cell, right? We have studied this in the cell subject. So the difference between the eukaryotic genomes and the prokaryotic genome is in eukaryotic genomes, the DNA first is linear, not circular. And second, it combined with a special protein called histone and to form a chromosome. And beside of the nucleus genome in here, in the eukaryotic cell, it have the other genomes, some scientists call it the second genomes, located in the mitochondria and the chloroplast. So as the scientists start to sequence the genomes of eukaryotic cell, they find out that the special organelles like mitochondria and chloroplast have its own DNA genomes. And this DNA is independently independent with the genomes of the host and similar to the genome of a bacteria. So they have provided a new theory called the animal system theory. We'll come to it later. So normally, the eukaryotic genome is linear DNA. It's different with the prokaryotic genome, which mostly is the circular double-strand DNA. It's compact with the protein called histone. In eukaryotic cell, in a molecule combine.
Sir,
Yeah.
I think you forgot to change the screen.
Sorry. Okay. Okay. Okay, so in a eukaryotic cell, the DNA molecule combine with a protein called histone. So, for example, this is a double-strand DNA. This is a double-strand DNA. And in cortex cell, it will combine with a complex of histone. So, 1, 2, 3, 4, 5. So this is called the octamer, which is composed of right up there, composed of 8 subunits of histone protein. Composed of 2 subunits of H2A, 2 subunits of H2B, 2 subunits of H3, and 2 subunits of H4. And the double-strand DNA will wrap one and the protein so the protein histone H1 is here. So this is the H1. Or we call it the linker. And it continues to wrap like this. Continue to wrap to another octamer, 1, 3, 4. Each octamer is 8 subunits, okay? Then continue to go around 1, 3, 4. Okay. So as a result, so you can see here the DNA strand wrap in here, and the histone H1 is called the linker histone. So it will twist another level to form a chromosome, and this chromosome is in the part tense is performed during maximally dense during mitotic phase in mitosis and meiosis. And this is the imaging image of the nucleosome structure. So of the each circle here is one octamer, okay, with 8 subunits of histone, and the string here is DNA fragment. So It looked like a necklace, a pearl necklace. Okay, was a combination between histone and DNA.
In this animation, we'll see the remarkable way our DNA.
Checking if I'm sure. Yeah.
In this animation, we'll see the remarkable way our DNA is tightly packed up to fit into the nucleus of every cell. The process starts with assembly of a nucleosome, which is formed when 8 separate histone protein subunits attach to the DNA molecule. The combined tight loop of DNA and protein is the nucleosome. Multiple nucleosomes are coiled together, and these then stack on top of each other. The end result is a fiber of packed nucleosomes known as chromatin. This fiber, which at this point is condensed to a thickness of 30 nanometers, is then looped and further packaged using other proteins which are not shown here. This remarkable multiple folding allows 6 feet of DNA to fit into the nucleus of each cell in our body, an object so small that 10,000 nuclei could fit on the tip of a needle. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope, chromosomes. It is important to realize that chromosomes are not always present. They form only when cells are dividing. At other times, as we can see here at the end of cell division, our DNA becomes less highly organized.
So with each histone subunit, it have a part here called the histone tail. So here each histone will have a small part put inside called the histone tail. And this histone tail is play a very interesting role during epigenetic that we will discuss in chapter 7. So the DNA is attacked with the histone to form chromosome, and the chromosomes was packing so dense, mostly in the metaphase during mitosis and meiosis. Much then is metaphase of mitosis and meiosis, so we can observe under the microscope. So let's come along with two terms of the chromosome is the heterochromatin and the achrochromatin. This is a two term refer to the state of the chromosome. The heterochromatin is referred to the state of the chromosome where it's maximally dense, and it is reached maximum under the metaphase. It's easier to for the cell to divide and split the chromosome under this state. However, for this state, because it in the mass dense, so the space inside the DNA molecule is not enough for the other protein can combine. So during that state, none of the process DNA replication or RNA transcribe can be performed. Instead of heterochromatin, it's transferred to another state called achrochromatin. While it's relaxed in the relaxed state, it can explore more space. So the huge complex like the DNA polymerase, which is not only RNA polymerase but also other transcription factor, can come and have the space to binding in the promoter. And like we discussed in the beginning of the eukaryotic genomes, we know that beside of the nuclear genome, each cell will have a second genome located inside the mitochondria, which is the mitochondrial genome. And in plant cell, we have the chloroplast genome inside the chloroplast. So this is come from a theory called the endosymbiotic. That means a very long time ago, based on any affected from environment. There is a symbiotic between the prokaryotic cell and the eukaryotic cell. That some prokaryote had come inside of the eukaryotic cell to maintain its function as a energy center for the eukaryotic cell. So let's watch a small animation to hypothesize how the endobiotic theory is happening.
Endosymbiotic theory explains how scientists believe eukaryotic cells developed. First, the nucleolus was surrounded by the enfolding membrane, creating the nucleus. Then further enfolding membrane formed the endoplasmic reticulum. Then the cell engulfed an aerobic bacteria, which would ultimately become the mitochondria. Afterward, a cell with mitochondria engulfed a photosynthetic bacteria. This photosynthetic bacteria would become the organelle known as a chloroplast. Mitochondria and chloroplasts contain their own small supply of DNA, which may be remnants of the genome they had when they were independent bacteria. It is important to note that we know that aerobic bacteria were engulfed first because there are animal cells which lack chloroplasts, while both plant and animal cells contain mitochondria.
Okay, so the next part is related to the human mitochondrial genome, which is, I think, is a very long part. So I will save it for the next Monday. Do you have any question on today's lecture?
Professor, I think there are some differences between the slide that you have here and the slide on Moodle.
Yes. So every year, when I doing my lecture, I have some additional, and I also delete some slide that I think is provide more information than necessary, and I added some another ones. But the main idea, the main backbone is still the same. So I will change the slide in the Moodle in the end of this course. Okay.
Okay,
But,
thank you.
The common, the backbone, the common as the same. The main part are the same between the slide in here, just as a minor addition. Okay. Another question, please. Okay, so if you have no more question, I want to thank you for your attention and see you on next Monday. Okay, thank you.
Thank you, teacher.
Bye, teacher.
Bye, teacher.
Goodbye,
Bye,
Professor.
teacher. Bye, teacher.
Thank you.